talk I'm really excited for is from Evan Macosko. So I've worked with Evan actually for a really long time, both at Harvard and now at the Broad Institute. The title of his talk will be Spatial and Single-Cell Genomics for Next Generation Neuroscience. And a little bit about Evan. Evan is an institute member at the Broad Institute of MIT and Harvard, where he directs a lab in the Stanley Center for Psychiatric Research. He is also an associate professor of psychiatry and neurobiology at Harvard Medical School and an attending psychiatrist at Massachusetts General Hospital. He serves as the co-PI for the BICAN Brain Cell Variation Grant. His lab develops new genomics technologies and deploys them to understand pathogenic mechanisms of brain diseases. They do some really cool stuff in his lab, I have to say, as somebody who helps him out with that. So let's welcome Evan to the stage so we can hear all about his talk.
Thank you very much, Tricia. Thanks, everybody, for being here. Very excited to talk to you about a large study that we've actually conducted very much with Tricia's help to systematically uncover all of the cell types, or close to all of the cell types that are present in the mouse brain. And I just want to mention that competing interest, I'm a founding member of Curio Biosciences, which actually is here today. The technology that I'll tell you that really drove this work, Slide-seq, to position cell types in tissues, has been commercialized through that company. So our lab is really interested in some keys under the lamppost problems in neuroscience, in which we think that neuroscience has suffered in some ways from kind of repeatedly focusing on and being unable to sort of comprehensively measure certain very key features in the brain. And the first one is brain cell types. Our ability to access and appreciate cell types in the brain is somewhat limited. We kind of repeatedly look at the same cell types over and over again in different behaviors and in different contexts. And this is for a variety of different reasons, but the main one is that we haven't been able to ascertain them comprehensively previously. And also, in the context of brain diseases, there are many reasons why it has been challenging to derive a clear understanding of how psychiatric and also neurological diseases arise. But I think one of the biggest challenges is associated with having good measurements, high-quality measurements of those tissues, and also ways of validating animal models that are related to those diseases. So just what my lab does is it tries to develop new technologies that allow us to uncover and tackle these particularly challenging problems. And then we use those technologies to build large-scale data sets that give us a bigger and more comprehensive picture of how the brain is structured and how it functions, and then we try to apply it to specific disease models. And I'm going to tell you particularly about this large effort to develop a comprehensive mouse brain atlas. It's work that we just released on bioRxiv a few weeks ago. But I also wanted to just mention a new technology that we also released on bioRxiv a few weeks ago that builds on Slide-seq that I think would be of interest to many in the audience, in which we've combined single-cell analysis and spatial analysis into a single protocol, single assay. And I encourage you to check out that work that was in collaboration with Fei Chen. It's, again, on bioRxiv, Russell et al. So let's dive into this comprehensive atlas that we generated. So the motivation here is that the brain does extraordinary things, and its outputs, are just kind of one of the greatest marvels of biology. But also, the other marvel is kind of at the micro scale, which is first appreciated by Ramon Cajal. Here's an image that he drew on the top. And also some more recent brainbow images that kind of capture the incredible complexity of cell types in the brain. This is not the way that other tissues of the body are constructed. There's just an incredible amount. Anywhere you look in the brain, there's an incredible amount of morphological and electrophysiological diversity. But it's been very challenging to sort of wrap one's head completely around this, to really understand all the different kinds of cells that are present. And to further understand how each individual region is composed, because of course you're kind of surveying a large organ, and it's unclear sometimes whether you're seeing the same cell type over and over again that may be just slightly repeated in a different context, or whether there actually is diversification that's occurring in these many, many individual anatomical structures. And so in our efforts to understand this cell type landscape in a comprehensive way, we wondered what we could learn about brain biology from this kind of comprehensive atlas. So today I'm going to tell you about the construction of this atlas and then 3 different sort of areas of insight that we derived from sort of understanding the principles of neurotransmission across the brain, some activity-regulated gene expression signatures that we uncovered, and heritability enrichment analysis that kind of points us towards particular cell populations associated with disease. So the driving technology for this project is twofold. The first is work, is high-throughput droplet-based single-cell RNA-seq, which began with our development several years ago now of DropSeq, which, really initially we performed. I know this video is not playing, but I just love to show. Here we go. I love to show this video because it's one of the first T-SNEs, which are now so ubiquitous in so many different talks. But this really captured our imagination. It's from the mouse retina, P14 mouse retina, and we appreciated just enormous amount of diversity that had not been seen before, and it really set the stage for being able to conceive of being able to do this across the entire brain. But the main missing piece here is where the cells are in space, and so together with Fei Chen's lab, we formed a really terrific partnership that has lasted over the last 5 years. We developed a second technology that utilizes the same barcoding strategies as DropSeq. It's called Slide-seq, in which you capture RNA locally at 10 micron pixels. And that technology allows you now to position cell populations in space. And this picture here is a piece of the hippocampus, but in principle, it was very easy to imagine extending this to the entire brain. And that's what we did. And actually, we say we were able to do it in principle, but of course the main way that we were able to do this was in really close partnership with several core facilities at the Broad. So the first thing we needed to do was to do millions of individual single-cell profiles from 100 different brain regions that an expert neuroanatomist, Chuck Vanderberg, microdissected. And we relied on Trisha's help to fact sort all of these cells to get very, very high quality, pure tissue. She actually loaned us a fact sorter into our lab so that we could do this every morning and get very high quality results. And then we relied upon GP to process all of these libraries, our genomics platform at the Broad. So this is what the entire brain looks like in T-SNE space. It's really just unbelievably complex. There are thousands of neuronal cell populations present, and that's just shown here by the color, which is purple for the neurons, and then all the non-neurons are in this smaller wedge here on the right. Now to position those cell types in space, we performed serial sections of Slide-seq. Over the course of a couple days, we generated 101 serial sections from a single mouse brain. That gave us 5 million individual pixels across that volume. And Carol Balderrama did an amazing job with this data set. It was so precise, and we missed one section out of 101 serial sections from a single mouse brain. We were able to reconstruct this entire, basically 3D volume of the mouse brain and register it to the common coordinate framework so we could know the anatomical position of every single bead within this large data set. And so now we integrated these 2 data sets to see where all the cell types that were defined by the single cell data were positioned in the brain from the Slide-seq data. So here is the initial mappings. This is the high-level mappings, and the first thing that really popped out to us is that the cortex is much less of a deal in the brain in terms of diversity than I think we might have expected. So in the darker green is hippocampus, and the lighter green is cortex. These are the areas that are the highest sort of focus of mammalian neuroscience. But this entire area here is below the telencephalon. So these are brainstem areas that are much less well explored than telencephalic areas. And now if we dive more deeply, these are each of these colors is one of the main brain areas, and each of these individual columns of this heat map is one of the microstructures, particular anatomical structures of the brain. There's like 300 that are defined in the Allen Atlas. And then each row here is one of the thousands of cell types, and you can see incredible specialization. Individual populations are found in just one or a couple individual regions. Some of these regions are enormously diverse. there are areas of the brain that have hundreds of cell types. So this area, the PAG, the periaqueductal gray matter in the pons, or I guess the midbrain technically, is an area that is involved in pain and fear responses. It has hundreds of cell populations inside of it. Whereas actually the cortex, we pretty much discovered everything that we knew that we would have expected to discover with just one individual assay. So, the diversity of the brain really does lie kind of in the brain stem, which I think is really remarkable and may not have necessarily been expected based on what we would sort of assume about the brain. So now that we have this data set and these cell type positions, we wanted to ask some fundamental questions about how the brain is structured. So the first thing is about neurotransmission. Every neuron utilizes some kind of neurotransmitter or neuropeptide to signal with other neurons. It's incredibly cell non-autonomous tissue. And so we used the gene expression to ask what kinds of neurotransmitters are used across all of these thousands of individual cell populations. So the first thing that we realized is that most neurons only use one neurotransmitter. They're either excitatory or inhibitory, and there's remarkable balance here, actually, where about the same proportion of cells are exclusively excitatory and cell types, and then the sort of reciprocal set of cells are inhibitory. And you can also see their distributions of all these different neurotransmitter populations across the brain. There are some very interesting and rare exotic populations, some of which have been studied before, some of which are novel, that co-release different neurotransmitters like acetylcholine, dopamine, noradrenaline, and serotonin. Then we looked at neuropeptides, because these are all genetically encoded, so you can just look at their expression. And this, I think, was a bit of a surprise to us. Basically, every cell type produces at least one, and usually more than one, neuropeptide. So your neuropeptide signaling is quite ubiquitous in the brain, and neuropeptide sensing is even more ubiquitous. So most cell types have at least 5, if not more, neuropeptide receptors. Now, most neuropeptides were discovered in the hypothalamus. So people kind of think of the hypothalamus as a place where neuropeptides are most important or maybe kind of exclusively found. But it turns out that actually every area of the brain is similarly sort of neuropeptidergic. They produce the same number of neuropeptides. What makes the hypothalamus and the midbrain, too, to some extent as well, unique is the number of unique neuropeptides. Peptides that are not found anywhere else in the brain are only found in the hypothalamus. So basically, these other neuropeptides are kind of recycled, like VIP and CART-PT. These are kind of recycled continuously over many, many different brain areas, but there's this kind of collection, about a third of them, that are only found in those couple areas. So most neuropeptide signaling is between neurons and other neurons, but we found some interesting examples of where neuropeptides, just by the fact that the receptor or the ligand is exclusively expressed in a specific cell type, seem to be signaling between neurons and glia. So one that we're particularly interested in is angiotensin. It doesn't cross the blood-brain barrier. It is produced in the periphery. But in the brain, it's only found, according to our data, in subcortical astrocytes. And then the angiotensin receptors are found in a collection of neurons. This is interesting to us because we recently found that the angiotensin receptor is the best marker that's been found for dopaminergic populations that degenerate in Parkinson's disease. So we think this angiotensin signaling could be interesting for that reason. So the second thing, I won't have time to talk about activity-regulated gene expression, but you can check out our bioarchive to look at that analysis where we looked at how activity differentially causes transcriptional changes in different cell types. But I wanted to just mention and conclude with an analysis we did of heritability enrichment. So there's an enormous amount of genetics data, human genetics data, that's been accrued for a variety of different brain traits. Diseases like Alzheimer's disease and neuropsychiatric diseases, as well as for things like educational attainment and many other kinds of traits that we would think of being related to brain function. And now we have this enormous and very comprehensive single-cell data set, or single-nucleus data set, that's now been positioned in cell types. We can utilize various heritability enrichment strategies, and we chose one from Alka Price's lab, to basically integrate these data sets together and ask which particular cell types of these thousands of cell types we found are preferentially expressing more of the genes associated with particular GWAS traits. And we chose to express this data as Manhattan plots. So you may be familiar with the Manhattan plots from GWAS data sets, where individual loci that are associated are sort of pop out of a chromosome-wide scan of SNPs, and you can see particular SNPs that rise to significance on a y-axis that's a p-value significance. Here, what we're doing is we're scanning across the entire brain using these localized cell types, and we're positioning so from left to right goes from anterior to posterior in the brain, and each of these individual dots is a cell type, and the y-axis is now the statistical significance of enrichment for genes associated with that trait. So you can see, for example, Alzheimer's disease shows a strong enrichment on these gray cells. These are actually the glia. We put the glia on the right because they don't have the same regional localization. This is kind of expected. We've known for a while that Alzheimer's disease has a kind of expression of genes that are enriched in glia, but we definitely see that strong enrichment in our comprehensive data set. But I wanted to focus particularly on schizophrenia because we found an interesting sort of dual enrichment in cell populations here in this blue population, which is the striatum, and also in the cortex. So when we look at these cell types, because now we know exactly which cell types are enriched, when we look at these cell types, we find that the cell types that are most enriched for schizophrenia GWAS signal are those that are found in the deepest parts of cortex. So these are cells that are in the layers 5 and 6 of cortex, and they're the cells that project to the striatum, which was interesting to us because, as I mentioned, the other population we found that was enriched was the striatal cell types, and we now looked more in depth at which cell types in particular in the striatum were enriched. So here we're ordering all of the cell types of a particular family that was enriched. They're called the medium spiny neurons. On the left are the most enriched, on the right are the least enriched. And then we first asked, because now we have all their localizations from the Slide-seq data, where these particular cells mapped to. And it turned out that they mapped to many different subregions of the striatum, both the dorsal and ventral striatum. Ventral is more involved in kind of habit and mood regulation, and the dorsal striatum is more involved in motor control. But it seems as if there was localization of cell types in these kind of highly enriched cell populations in both areas. And so now we could kind of overlay gene expression that's been known to subdivide these cells further by different pathways, the direct and indirect pathway. I can't get into all of these individual details, but the particular thing that stuck out to us is that a couple years ago, we identified a new population of striatal cells from single cell data that had not been appreciated before. It's called the ESPNs. And when we look at the expression of ESPN markers, we find that most of the ESPN markers are the ones that show the highest enrichment for schizophrenia signal. And so we're very interested in this idea that perhaps signaling between these cortical cells and these medium spiny projection neurons may have some important roles to play in the pathogenesis of schizophrenia. So in conclusion, we've developed this large-scale brain atlas that we think provides some really key insights into the diversification of the brain, which is just enormous and baffling. We still don't really understand why there is so much molecular diversification of the brain and what this actually means from a functional perspective. But we've really appreciated that the brain stem in particular is a home to a lot of this diversity. And you might think, well, what is so special? What's interesting about the brain stem? Well, it's been implicated in a variety of sort of basic functions like physiological autonomic regulation, but it's also a site of active evolutionary innovation. And so I just wanted to highlight some work from last year from our lab where we did a comprehensive survey of midbrain dopaminergic neurons in the rodent and primate and human. And we found a dopaminergic population that was unique to primate. It's one of only 2 examples in the literature of primate-specific cell types in the brain. The other one is also not in the cortex. So we think that brainstem is not only sort of an evolutionarily sort of conserved and kind of basic structure, but it's also being actively sort of diversified even now in evolution. We also found that peptidergic signaling is a ubiquitous property of neurons, and and we've developed this way of connecting gene sets to particular cell type groups. So we've made all of this data pre-publication available at a website, braincelldata.org. It allows one to plot all of the expression of genes within the Slide-seq data. You can position cell types there. You can see the expression in cell types of every gene. And you can do a variety of different kind of simple analyses to look for genes that are enriched in particular cell types that are enriched in particular regions. So there's a lot of different things that one can do there to explore this data, as well as just download it if you're interested. So I think I will skip this, but the next step is we have to figure out what all these cell types do. There's really no playbook for doing this, and Faye, my close collaborator, and I are really trying to figure this out. We have to find a systematic way of going through the brain and figuring out what these do, because the mystery here remains. Why does the brain do this? And we're very interested in exploring this in the future. So with that, I wanted to just acknowledge Jonah Langlieb and Nina Sachdev, who led the analysis of the Atlas work, which really was heroic, acquired 21 computational years of time to actually compute all of these individual analyses, and also other members of the Macosko lab and also the Chen lab that worked together to make this analysis possible. Happy to take any questions now or later. Can we take any questions now?
We have time for a few questions.
Yeah.
Can you come to the microphone? There's one right here.
Hi, so thanks for the talk. So my question is pertaining to the neurotransmitter part of your talk. So I do understand that you can get a pretty good understanding which cell secretes the neurotransmitter from the transcriptome data itself. But there are few publications now that can actually capture the secreted protein with the transcriptome. So I was wondering what your thoughts are on whether adding that modality with the transcriptome, like looking at the secreted protein, like looking at the neurotransmitter themselves, can add any value to the data, and if yes, how?
Oh, that's a great question. So secreted protein analyses would be particularly valuable for neuropeptide signaling. So neurotransmitters are small molecules, so they wouldn't necessarily be suitable for that. But for neuropeptides, I think neuropeptides represent an incredibly confusing and currently technologically intractable mode of communication to study. They don't seem to be released exclusively at synaptic terminals the way that neurotransmitters are. We actually don't really understand all of the ways in which their release is controlled. So certainly being able to simultaneously understand a cell's state and it's also its actual secretion of neuropeptides, and particularly where along the cell a particular neuropeptide is secreted, could be very valuable, because we think of neuropeptides in the sort of traditional way of neurotransmitters, that they're kind of secreted at a terminal and maybe they modulate the neurotransmitter's activity. But there's a lot of examples actually where they're secreted in the soma and they maybe act more like a neuroendocrine kind of system as opposed to a neurotransmitter system.
Thank you.
I think we have time for one more question.
Fabulous talk, thank you so much. I'm just curious to know, with the new approach you are taking, have you been able to look at cell plasticity and addressing long-standing questions about regeneration in the brain, whether you saw new phenotypes that had not been looked at earlier?
So in our data set, we do see the traditional places where we know neurons are regenerating. So we see cells that are going through, neurons that are going through cell cycle and differentiation in the hippocampus, which is the dentate gyrus, which is the way where we've known there to be generating cells. And we also see the migratory stream, the dorsal migratory stream of cells that are moving towards the olfactory bulb, which is another area of active neurodegeneration throughout life, regeneration throughout life. We don't see any evidence of any other cell types in any other part of the brain undergoing those kinds of transitions. So we think that probably those are the only two areas that are continuously regenerated.